Good morning. I am doing a dress rehearsal today for training that I'm going to be conducting in Jena next week. It's intended that you should be able to see these trainings on the first day of the show at GJAM, but we had more people sign up to take part in the training than we have space for in the room. We have table space for about 30 people, and something like 61 people signed up, which was a lot more than I anticipated. So I'm really glad that you were able to come here today, and we'll walk through the demonstration and do two really kind of different takes on what proteomics is between the morning session and the afternoon session. This morning we're going to be talking about what I consider the bread and butter of proteomics. I have a bunch of examples, a bunch of replicates of type A, a bunch of replicates of type B, and I want to know what's different between them. What's going to be a little unusual about this is that we have data from what's called a DDA style experiment and a DIA experiment. Now, if you've been in proteomics a long time, that probably makes perfect sense right from the word go. But I don't know how broad your proteomics experience has been in the past. So who has looked at data from a proteomics experiment or had some training in class? Okay. Proteomics doesn't always make friends right away, so I understand. Okay, so I will not start with slides because I really feel like with 2 hours we need to emphasize doing rather than talking. But I'll start by saying that a tandem mass spectrum, or an MS-MS, is the death cry of a peptide. I know that's a silly way to put it, but when a peptide is isolated, it can then be given a little voltage to cause it to move about more aggressively. It gets energized, and it dissipates that energy by breaking a peptide bond. Okay, so a tandem mass spectrum is recording the fragments produced when a particular peptide gets blown apart. That's how DDA, or data-dependent acquisition, works. DDA focuses the attention of tandem mass spectrometry on the peptides that are currently available coming off of the LC column at a given moment in time. A given proteomics experiment typically lasts something like 90 minutes. So it's often the case that you have what we call the shallow gradient, a shallow rise in hydrophobicity, causing the peptides to come off of the column and flow into the mass spec. So the instrument is choosing to sample what peptides create intense signals in MS scans and selecting those for fragmentation in tandem MS scans. So we're going to have essentially three processes that come into play for FragPipe to handle. And those are, first, identification, second, filtering and assembly, and thirdly, quantitation. The identification is the process of figuring out which peptide corresponds to a particular MS-MS. That's handled by the software MS-Fragger. The second phase of that is figuring out which spectra have been successfully identified and which unsuccessfully. So that's the process of filtering. It's also going to use that a variety of methods to figure out which proteins these peptides belong to. That's protein assembly or protein inference. Then that's all handled by the philosopher tool, and the philosopher tool is why this won't run on a Mac. It's not all the tools within philosopher have been ported to run equally well on a Mac as on a PC. Then the final step is quantification. How do we say how much of a given thing is there? And that is handled by ion quant in the case of the DDA experiments. So all of that is being wrapped up in one big workflow by FragPipe. After that, we're going to poke into a website called FragPipe Analyst that will do statistical testing to say what actually changed between these cohorts. And then we'll repeat this process, except this time with a mode of mass spectrometry that's much better at quantifying everything. That's the promise, at least, of DIA. So we'll talk about those differences as we move along, but I didn't want people to be taken from 0 to 60 and assume that you know all the steps in between. There are flash drives passing around. There should be 4 of them. So if you don't have a copy of the data for today's demo, please grab a copy of one of those drives. Okay, I am not intending to go through the process of installing FragPipe. Hopefully you've already done that. I would mention I'm already using an old version. This is from about a year ago. The newer versions, FragPipe 23 and later, have gotten a whole lot easier to install because they take care of getting Python for you, of getting Java for you. And in the past, I had to argue with which specific version of Java was necessary to making the software run. So I've gone through that pain here. You don't have to. So what are we looking at? When we start with the FragPipe user interface, you might ask, what is FragPipe doing in all of this? And the answer is FragPipe is coordinating it all. It's making sure that the outputs from one step are written in the correct output format for the next one. It's helping to automate what are a large variety of workflows that we'll talk about in just a moment. And what you can see on this first interface is just how many different software tools are being tied together. Up at the top, we have MS Fragger, IMQuant, and DIA Tracer. This page in particular looks different on version 23 than on 22, but the impact is quite the same. So we can see then that we have all of these little tools that have been installed along with FragPipe to do all the work of identification and quantitation. If your software says that these are not yet present, you're going to have those installed. So you need to click through all the licenses and downloads to get those in place. Okay, we have some software called Diane in the next block here. Diane is external software made use of by FragPipe that is quite necessary in the DIA data analysis step that we'll do at the latter half of this morning. We have some software tools for Python. Python and Java are the two principal languages in which the FragPipe tools are encoded. So we need to be able to have a reasonably up-to-date Python, but not too up-to-date in this version where the software won't work anymore. It's crazy. So I'm really glad that Python is now installed for you when you install FragPipe.
Yeah, I think I'll have to separately install because I have the NA for all of the...
It'll be the same.
But I think if you click downloads next to it, then you have to register as an academic account or something. That should be freely available.
Like the top one.
Yeah.
Yes. Yeah. So So if you need to get these tools in place because it doesn't show versions, the download and update feature should take care of that for you. Yeah.
But you will have to fill in a form and get a verification email sent to you.
I don't think it requires a verification anymore, does it? Oh, does it? Okay.
But if you put in your working account or other educational institution, they will send a link to that.
Yeah.
And it's like a 6-digit code.
Okay. All right. There are some advanced features that can be used, especially in scenarios where you have a very large database of peptides you need to compare to. Database splitting can be useful for that. In a case like that, there are certain libraries that need to be installed in Python for that to be feasible, pandas or NumPy or something. That, I think, is handled automatically in the newer versions and not in my older version. And this last part, the spectral library generation, is not going to be necessary for the first part of this morning, but it is necessary for the second part of this morning. In the old days, installing this script properly was quite a nightmare. Again, that's gotten a whole lot better in the most recent editions of the software. In this case, when I click the Install or Upgrade EasyPQP button, it appears that nothing is happening, but the computer is actually laboring away, and 2 or 3 minutes later, I'll get a screen full of garbage that is all to say it worked. So all of those are going to be needed this morning. All right, so I'm going to move on to the next tab. We'll kind of be working our way from left to right every time we run this software. So we start with the config to make sure everything is in place. I mentioned that there are a wide variety of workflows available to us within FragPipe. The one that we're going to use in the first case is the LFQ MBR option. So if you've been in the proteomics world for a while, that probably makes sense, but for new people, I don't expect it to. LFQ stands for label-free quantitation. So it's commonly the case in proteomics that if you're quantifying something, you buy some expensive reagents to, for example, tag each of 4 different samples with a different label on each of the peptides, and then you use that for quantifying more accurately. But in general, we find that label-free quantitation is the most common way that people do quantitative proteomics now, which means integrating the peptide signal without any isotopic labeling at work. And that's the LFQ part. The other is MBR, or match between runs. This is the This is based on the premise that a peptide may not be identified in multiple runs, but it may still be detectable in intact form in the MS scans. And so match between runs allows us to quantify peptides in an experiment, even if they haven't been identified there. So LFQ and MBR are quite necessary there. I believe in the new version, you no longer have to click load workflow, but in the older versions, if you don't click load workflow, it doesn't actually apply the change. So I'm going to click my load workflow button. All right. How expensive do you want this to be on your computer? If you're willing to use all of the RAM you've got, then specifying 0 is fine here. Parallelism says how many processor cores are you going to allow it to use? This laptop has 8, so I'm going to say 8. If you know how many processor cores you've got, you can specify it there. Or if you want to keep working on something else while this is going on, you can make this fewer than the number of cores you've got, and then you'll have a CPU saved for you to do other stuff at the same time. All right, the data we're going to use are regular MS spectra. They're not from an instrument that features ion mobility. If you were working with data from the timsTOF on the 6th floor of our building, you might be using ion mobility, and you would need to specify that option instead. Now, it's worth saying that the files I've given you are not quite ready for your use. If you go into this Part A cohort, these are of the files that you've copied from the thumb drive, you can see that the data I've provided in MZML format are all zipped up. The software can't read them that way, so we need to unzip them. The simplest way to do that on most computers is, on most Windows computers, is to use the extract all option. I use 7-zip because it's a lot faster, but I'll just use extract all here because it will work. All right, so it's going to take that one large file and explode it into 6 larger files that are uncompressed.
Extract. Yeah.
You are brave trying to shift away from using a Mac for this morning's class. That is it's a learning curve. It's a learning curve.
Yeah. I checked.
Okay, when I ran extract all, I got a new subdirectory called DDA MZMLs, and you can see that there are 6 files inside. If I can take just a moment to talk about the experiment that produced these, I think that will help give us some context. We want to do a comparison of a whole bunch of samples of type A and a bunch of samples of type B, and we want to say what's different. In this case, the data were synthetically made. They have a yeast background, just all the peptides of yeast, and into that they've spiked something called the sigma UPS2, which is a mix of human proteins that have been added at different concentrations in samples of type A and samples of type B. So what we're going to look for is our ability to detect those differences, the human protein differences, between these 2 types of samples. The file names give you some indication of what's going on here. We have some that are listed as ratio 0.1 and others that are ratio 10. That tells you that there's 10 times as much of the human protein spike in the ratio 10s as there is in the ratio 0.1. Okay, so now that those are unzipped, I can return to FragPipe and tell it I want to add those files to this run. So I need to find them. In my case, I shove them on D drive in GJAM training Part A DDA MZMLs. Now, your path is quite likely different than that. And I will note that sometimes FragPipe gets very fussy if you put the files in a directory that has a space in the name. The reasons why are not important, but that is something to be aware of, and it's one of the reasons why I put them here rather than on My Documents, for example, because the directory My Documents has a space in it. Okay, so here I can simply click Select. What did I just do? Oh, I didn't actually select them? Oh Lord, that's a silly mistake. Okay, yes. So having told it, yes, I actually want the files in that directory, not just to show me the files in that directory, that's good. So now I have these files here. Now you can see it's already truncating the file names because that column is not quite wide enough. Let me pull this a little wider because it's going to tell me which files belong where. Okay, it's way over there, isn't it? So the path is already quite long. And then over to the right, I can now see ratio 0:1, ratio 10, and so on. Now, it's not necessarily important to FragPipe this next step, but it's very important to what we're going to do in FragPipe Analyst. We need to specify what cohorts these 6 files belong in. That's why we needed to see the file names. So in this case, I'm going to use the experiment label 1 and the experiment label 10. You can see that this is a ratio 1 file, that's why it belongs to the 1 cohort. This is a ratio 10 file, that's why it belongs to the 10 cohort. So for each of the 6 files, I need to specify which cohort it belongs to. All right. And for this last thing, I need to have some numbers that specify how these things relate to each other. In this case, each raw file, each MZML file in this case, is a different experiment. In some cases, you would use multiple raw files to explain all the peptides of a particular sample. We're not working with anything that complex today. So we can just call each of these a different bioreplicate number, and everything is happy. So for now, I'm just going to call this one 1, this one 2, this one 3, this one 4, this one 5, and this one 6. So it's not particularly subtle what's going on there. I'm giving arbitrary names to these 6 files. If I wanted to make it easier to trace back to which file produced which identification, I would probably use something like these numbers because they change for each file, 248, 264, 272, and so on. I'm just not going to take the time to do that at the moment. It's helpful in that case to tell you which file corresponds to which replicate. All right. With that, we're done with this page. Does anyone need me to pause before moving ahead?
Yes, please.
Oh, sure. So a natural question that you might ask is proteomics limited to my comparing a cohort to another cohort? And proteomics is capable of doing much more than that. You can certainly have experiments with 3 different factors and cohorts of irregular size and so on. You can do stuff like that. I'm choosing a simple example of comparing 3 runs to 3 runs because this is far and away the most common way that people do differential proteomics. I would also say that software like FragPipe Analyst, the statistical framework we're going to use to look for these differences, is really designed for 1 cohort versus 1 cohort. The software is not designed to do modeling of multi-factor studies, for example. Just to give an example of that, some of the people I worked with in South Africa enjoyed working with multiple species to evaluate their resilience under different climate conditions. So they might have a well-watered turf in which they were growing their plants, so they might have drought-like conditions, and they might have 2 different species that they were trying to compare differential responses in. In a case like that, you definitely do need to do a multi-factor study. But in this case, we're just doing a bunch of A versus a bunch of B, or a bunch of 1 versus a bunch of 10s. Any questions before we move ahead? Okay. Now, we see that there is a DIA pseudo MS2 step, but we are not going to use it. This is not a DIA experiment, and we're not going to use the DIA umpire approach anyway. So we just skip past that. But on the database page, we need to specify a search space. Okay, so in this case, the software needs to know what protein sequences are real. What protein sequences could these peptides plausibly be? We don't consider the space of all combinations of amino acids. That's way too big and nasty a space. Instead, we're going to consider just the protein sequences that are in yeast or in the human spikes. If we return to the files that we copied from the thumb drives, you should see something right here that is a FASTA file. This one already has decoys added to it. It already has contaminants added to it. It already has the IRT peptides, and it has the UPS added to it. So that's a whole lot if you've never seen one of these before. First off, we call this a sequence database. And a lot of people would assume then that a FASTA file is actually a very complex thing to deal with. The reality, however, is that FASTA files are just big text files. So I'm going to open a text editor and throw this FASTA in there. All right. The font size is definitely too small here, so I'm going to improve that a little bit. I want console, I want about 18 point. Since turning 50, my eyes have told me things are a little different now, so I have to be a little more cognizant of everyone's vision.
I feel you.
It's weird, it's not showing, it's cutting off the left edge of the screen a little bit, so let me pull this in a little. Okay. So FASTA databases are big text files. They have lines like these that say, hi, I'm a new sequence. So this greater than, if you see a greater than symbol at the start of the line, it's giving you the accession for what this protein should be called. It's got some sort of description that gets tacked onto the end of that. And then the rest of this is several lines of sequence. Okay? Those are the chains, literally the chains of amino acids that comprise this particular protein. The software is going to extrapolate from these protein sequences what peptide sequences could be observed for each protein, given a certain type of enzymatic cleavage. Here we use trypsin. So we provided protein sequences. It turns those into peptide sequences in silico and compares those to spectra. Is it possible to create your own sequence database? Yes, absolutely. But as I mentioned, this one has gone through some steps. Well, let's start with the fact this UP number right in the middle. Do you remember I said that these are human proteins spiked into yeast? Right. So UP00002311 is the baker's yeast reference proteome downloaded from UniProt. So this is their canonical name for every protein in yeast. IRT peptides are sometimes used to make sure that the chromatography is causing very similar separations of the peptides with respect to time. So we added those in, the IRT peptides. If you want to see what those look like, you can look at source FASTAs. The UPS proteins, those are the human proteins that got spiked in. But it's very important for us to be able to control the false discovery rate of identification. And for that, every protein in this database appears in two forms. It appears in the normal orientation as downloaded from UniProt, and we reverse it, N-terminal for C-terminal, and add that as a decoy sequence. Okay, so we're going to compare each peptide or each spectrum to twice as many peptides as are real, just as a way to control our false discovery rate. That database is what we'll then supply to FragPipe for the search. Now you might say that sounds like an awful lot of crazy manipulation to make all that work. And helpfully, you can actually do a lot of this work in the graphical user interface. You can see it already has pre-baked sort of the most common reference genomes that you could grab, but you can specify your own on the command line if you're working in a species that it doesn't have listed there, and that's fine. Typically, I tell it to include reviewed and unreviewed sequences. I am probably the lone voice in the wilderness who feels that way. But if you want to ensure that every protein coding gene of a species is included in the sequence database, Gesundheit, you need to turn off reviewed sequences only. Adding decoys is necessary to controlling the false discovery rate. Oh, I'm sorry, this was from the download button on the database page. Sorry. We can tell it here to add decoys so we control the false discovery rate. Adding common contaminants is quite necessary because very frequently, even though you're working in yeast or slime mold or C. elegans worms, You're going to find human proteins anyway because we are humans and we release a lot of proteins into our environment. It's quite commonly the case that you'll find keratins from your skin in there. And frankly, if you're wearing a wool sweater on the day you're working in the lab, you can find sheep keratin too. So these experiments are really, really sensitive. All right. Yes, sir. The IRT peptides are important in cases where you're trying to make absolutely sure your liquid chromatography is nice and standard across the experiments. IRT peptides get spiked in to represent kind of landmarks in retention time. So I don't think we do. Does anyone on our site use IRT routinely for experiments? Not the core facility, not you guys. So it's not so popular here, perhaps, but yeah. If you're trying to make sure your chromatography is bulletproof, you stick in the IRT peptides. Yeah, it's not a huge issue or necessarily commonplace. It might be that the question you're asking biologically requires knowledge of which particular isoform is present for a protein, because there are different messenger RNAs you can produce from a protein coding gene. Yeah, so I'm going to cancel out of this. I wanted to show you the downloading interface because it exists, but we already pre-made the database. So take this as context, basically. So let's go find the database in the right location. It was on my drive D. Oh, gosh. My drive D. And it was in the GJM training. It was in part A. And then it was this one, the Valentine's Day database. Am I the only person who does a lot of non-romantic stuff on Valentine's Day? I wrote this tutorial on Valentine's Day. Okay. So we're going to just grab that database and go with it. Okay. So we're done with the first 4 tabs. That's all good. Let's get to the identification step. This is a place where you can go hog wild. I mean, there's all kinds of options here that can modify how we go about doing the identification step, because that's what MS Fragger is all about. And for a long time, when people said, I need to do database identification, they were just talking about the database step. The pieces after this actually take just as much time as identification does now. But there are some broad things that we need to talk about here. Maybe you're working with older data that were captured, say, on an Orbitrap Velos, and that might have very accurately measured precursor or peptide ions, but very inaccurately measured fragment ions. And in a case like that, you would need to change the fragment mass tolerance to reflect that you have low-res stuff. Most of the instruments that we would use here, the Exploris, the timsTOF, you can use these default settings just fine, and everything is good. What if you used something other than trypsin? You might need to change away from the strict trypsin definition and instead specify that you use trypsin but added a little bit of cyanogen bromide, or used chymo or whatever. That's possible here. You can use combinations of digests if you like. All right. I think talking about variable modifications is important, so I'm going to pause here for just a moment. This is something that's going to come into play later in the day, but it's quite commonly the case that if you have peptides with methionine in them, you look at them a little squirrely and they get oxidized. If those peptides are oxidized and you don't allow the software to take that possibility into account, you would just misidentify those peptides or fail to identify those peptides. So it's very commonly the case that when we run a search, we allow for methionine to gain 16 mass units. It does not require methionine to gain 16 mass units. It allows for that possibility. Similarly, we would look for a gain of 42 mass units. And here they've specified that the site where that should be is square bracket caret, which is not necessarily an obvious term. So if I hover the mouse here, I see that square bracket is a protein N-terminus, and that caret just means on the terminus, not on the side chain itself. So why would we expect the most N-terminal residue of a polypeptide to gain 42 mass units? We said that plus 16 was oxidation, right? So plus 42 on an N-terminus is what? Protein biochemistry question. You didn't realize that was coming. Okay. Yes, it's acetylation. Excellent. Nothing like looking up the answer in the back of the book. Yes. Yes, it's very commonly the case that the N-terminus of a protein carries a plus 42 mass shift. So we add that possibility here. But there's a little bit more to talk about on that score, and it's right here. These are fixed modifications. They are a different class than the variable modifications. Remember that I said we will allow methionine to gain 16 mass units? That's a possibility. But here we are requiring that all cysteines have a mass shift of 57 on them. This is very commonly the case in proteomics, and yet sometimes that's not the right mass to add. This is coming from reduction and alkylation of disulfide bonds. So we cut the peptide bonds with
DTT, yeah.
DTT, yes, most typically, and then we would treat them with iodoacetamide. Iodoacetamide or chloroacetamide, either way, would lead to a mass shift of 57 on cysteines. And that reaction runs almost perfectly to completion. So we would expect all cysteines to be modified in this way. I got to check my time because I'm going to run myself into trouble here if I keep talking this much. All right. There's a lot of other stuff you can do here. We'll talk more about modifications in the afternoon. All right. Any question? Yeah. Oh, okay. Yeah, so we didn't end up making any changes at all on that MS Fragger page. We see that you can do lots of things there. We're not going to do them. We're going to restrain ourselves to standard mass shifts at the moment. The validation tools have a lot of impact, but in effect, most people leave these mostly alone. There are a lot of steps involved in deciding that this spectrum has been identified and that this collection of spectra represent these peptides, and these peptides represent this protein. That's what this page is really all about. And you can see that there's a lot that's going to take place and a lot of named tools for this purpose. MS Booster, Philosopher, Percolator, Protein Profit down here, all of these are going to get involved in the same process. We'll see them as they run. But at the moment, we're not going to make any changes in how those are to be configured. PTMs, we don't have anything really exploratory going on here. We leave this page alone. Glycosylation, we're not messing with glycosylation today. But quantitation, we are. Remember that we said this is an LFQ MBR experiment. Did you get a copy of the files?
I don't think so.
Oh, okay. We've got a flash drive moving around with those files. Excellent. All right. I got to get those back at the end of the day because I have to hand those around for the students on Tuesday. Okay, so IonQuant is software that's going to attempt to say how much of this peptide is present by integrating the chromatogram that we see in the MS scans. Essentially, the MS scans are a way to say which ions are coming off the column at this moment in time. A given peptide is probably present for around 30 seconds. So ideally, you will have several MS scans cutting across that allow us to quantify it. Okay, again, we're changing nothing here. All right. Isobaric quantitation is not in play. We're not using labels of any sort that allow us to do that. We don't need to worry about spectral libraries. We don't need to worry about quantifying in DDA mode yet. Skyline we're not going to mess with, so we go directly over to the Run tab. Now here, I need to specify where I'm going to save my result. So once again, what's that? Oh, sorry. Okay, so I'm going to go back to where I installed the files on drive D, GJAM training, part A, and I'm going to just create a folder in here, and I'm going to call this training run 1. Maybe I'll even call it DDA. Training run 1 DDA. All right, so I can select that folder for writing my outputs. I don't want it to completely spam my hard drive with a bazillion files, so I'm going to check the delete buttons here. This again looks slightly different on version 23 and 22. Any questions for folks?
Yeah, we also. Oh, sorry.
I'm sorry?
I have from this page, just froze to that's it.
Yes, you just provide it on the database page. You just specify the database that was on the thumb drive. Yeah, that looks correct. Yes. Okay. So with that, we should be ready to run. No? Oh.
I'm just curious if it's going to.
I already did something.
Oh, okay. Oh, you already hit the run button.
Well, I don't know if I
Oh.
did. I have it.
Yeah, okay.
Okay.
It's just Python being mouthy. It is that way. All right, so I'm going to hit the run button and see what happens. Now, ideally, you're getting a whole spray of text showing up on screen as the software starts working its way through the problem. I often like to see what kind of load this is putting on my system. I'm going to come down here to the taskbar, right-click, and say Task Manager, because it's useful for that. Then I can click here. All right. So we can see that it's definitely taking some CPU time as it does this first step, but probably for most ordinary laptops, the memory load is going to be the biggest issue. If you have a laptop with 4 gigs of RAM, this is going to be pretty painful in all likelihood. Is it behaving itself on 8 gigs?
Which one?
Click on the memory. Yeah, it's using 6.8 of 8 gigs. That's
7.8 even.
that's a good life. We're making it forward.
I got insufficient memory.
Oh,
And
okay. Very frequently, it's the web browser.
Yes.
If you have lots of tabs open in your web browser, that is going to be a problem. There are lots of reasons why you can run out of RAM, but for me, it's almost always the browser. So.
I just put run again.
Perfect. It probably complained about how there might be files in the directory, but that's fine.
Okay.
You can overwrite them. So it's doing a pre-search in this first case. The software wants to sniff the data and make sure that the mass accuracy that it finds is representative of what it expects. If all of the peptides are 4 ppm off where they should be on the mass axis, the instrument was miscalibrated, and the data will look really weird unless the software is able to recalibrate them to take that into account. In this case, we see that at base, we typically were about 1 and a bit ppm out of true on this data set, so it's able to correct that so that it's spot on. In some cases, you will find data that are, up to 10 ppm out of calibration. If they're worse than that, the software will choke on it. Actually, it'll just say, "Forget this. I can't deal with it." Right. So there are several parameters that can optimize in how to do this search, and it's doing that to increase the fraction of the spectra that it can explain properly. Remember at base that we're asking the software to say, what peptide sequence does each tandem mass spectrum represent? Okay, and with that, it's into the main search. It's got these 6 files, and so each one of those needs its thousands of spectra compared to peptide sequences. Is this running on everybody? No? Insufficient memory. Okay. What does Task Manager show you? Click on the little heartbeat symbol and then click memory. Yeah, something's using a lot of RAM. If you click on that one, and then sort by memory, you can see what's taking up all the RAM. Yeah. There's a Java job that's using the RAM at the moment, so I think that Java job is probably MS Fragger.
Probably.
Okay, but did it stop itself when it had the memory error? Oh, okay. Let's go back to MS Fragger. Oh.
Yeah, the processes.
Yeah, it says it stopped running.
Yeah, maybe do you have important things in Microsoft Edge open?
You can, but it'll close MS Fragger itself. It's strange that
If you're running low in memory, you could also try splitting the database.
That's true. I think that decoy is enabled by default.
Decoying,
I don't think it's
yes, but
finally
if you split the database, it runs multiple searches and then concatenates the output at the end.
Okay.
So you need less RAM to run the individual search.
Yeah, is that option covered on the MS Fragger tab? Oh, split database right here, sure.
Okay. Increase that to like 4.
Yeah,
You need
that would make it do the search in multiple passes. Yeah.
Sorry.
Oh, this is this is on the MS Fragger page itself. Good suggestion.
Seems to be a lot of resource issues. So
Yeah.
Let's do.
I have to say that as a bioinformatics person trying to gauge what kind of hardware, trying to gauge what kind of hardware your users have access to is a very difficult thing to do. When MS Fragger was first developed, somebody with 16 gigs of RAM had something special, but not anymore. And they just expect that you're going to have a fair bit of RAM on the computer. So yeah, in my case,
Maybe it's also good to mention if people have a profile data, in the Orbitrap.
oh yes, in this case, the data are MZML format. So I've already reduced the size of the raw file by peak picking it to make the MZML files.
Just for the others, maybe if they have a Orbitrap profile, and they have if you do not knowing how to convert it to central.
Yeah. In general, if you give data to MS Fragger, the simplest way to do it is just to use the raw files as produced by the instrument. Because this is a tutorial on handing around the class on a thumb drive, I try to keep what I hand you very, very compact. And in my case, shrinking the data by peak picking them and writing them as MZML made it a lot easier to hand those out on a thumb drive in a very compact format.
And out of curiosity, how is this analysis that we are doing on FragPipe different for example, what the mass spec facility provides us?
Very frequently, the mass spec core facility is going to produce DIA type experiments rather than DDA. So the latter half of this morning will be more relevant to that. They don't use FragPipe in their data analysis. They're almost always using Spectronaut, which is very much designed around DIA data analysis. So Spectronaut has roughly the same kinds of questions you would ask in order to launch the software. You still provided a FASTA sequence database to say these are the protein sequences that are possible. It still has to do some mass recalibration to tune it up. But the way that data from the core get quantified is a little unusual, frankly, for DIA experiments, because they rely very heavily on MS1 scans for quantitation rather than making use of chromatograms from fragments. We'll talk about that some more as we get into the later part.
Yeah.
But you can ask, how to set up your experiments. You can ask if you want DDA or DIA. So if you know the difference and you know why you want to have DDA, you can just ask to have a method with DDA.
At the moment, I would say the number one reason I would ask for DDA is that I intend to do post-translational modification work in my data. If I'm just trying to quantify broadly, using DIA spectra is very preferable, I would say. So anytime you're relying on the quantitation, the DIA is likely to come at some advantage. Is it still barking? It's doing something. Okay, well, maybe it will run. I mentioned that this workflow is not going to be limited to just the problem of identifying spectra. It's also got to figure out which of those spectra it actually believes, and that's what it's working on right now. It's using an iterative process to figure out the best way to separate good peptide spectrum matches. That's what this PSM stands for. The good peptide spectrum matches from the unreliable ones. Okay, so percolator and booster and protein profit are just going to keep rolling by doing their thing. But I would point out that this is a little challenging because it's not just our data are spread across 6 experiments. And the same peptide may be observed in any of those 6 raw files or 6 LC-MS experiments. And you may even have multiple spectra identified for a given peptide within the same experiment. So at present, the way that most people in the proteomics field deal with identification, they assume that each MS should be evaluated independently. So seeing other spectra for the same peptide gives that peptide no advantage in identification for a given MS. But at some point, the software has to rise to a higher level and say, given this set of spectra that I think are confidently identified, which peptides can I say confidently exist in this sample? From that set of peptides, how can I infer a parsimonious set of proteins that explain the existence of those peptides in the sample? And we're trying to do comparison across experiments as well, which means we need to have one protein list in common that spans all 6 of the experiments. So those are the kinds of things that are happening at this stage. And now we see that we're into the filtering stage to actually apply the rules that the software has inferred for separating the good from the bad PSMs.
But does it mean that you are expecting that all 6 samples should all have the same identified proteins in different levels, in different amounts?
It is acceptable to invoke a protein because it has enough evidence in these 3 files, even if it's got no evidence whatsoever in the other 3 files.
Okay.
So it is not making the requirement that peptides be seen ubiquitously, or that the evidence is spread evenly across the data. It's
Okay.
but it does need to have one inference that explains that I guess I always think of things as kind of a big spreadsheet. I'm going to have a column for each of the 6 raw files, and each row is going to be some protein. It's okay if a protein has 3 reported intensities for the first 3 replicates of cohort 1, but no intensity reported in the other 3 because it's a different cohort. That's what we're looking for.
Yeah, exactly.
That's a biomarker for us.
Yeah,
So,
yeah.
yeah. All right, finally it's gotten to a quantifying step. So we finally moved into the ion quant stage. So ion quant is looking at the first appearance of a peptide ion in an MS1 scan, rising to a crest as you get a couple seconds into its moment of arrival, and then it will very slowly sort of fade, tail off at the end. It needs to quantify that curve of intensity values seen for the peptide, and it writes that down for each raw file separately. All right. I have to say this is a very big change from the way that protein identification worked when I was in grad school. We had a does anyone here remember a Pentium Pro? The Pentium Pro.
Yeah.
Yes, we had a Pentium Pro, I think at 200 megahertz under Jimmy Eng's desk. And we ran Sequest compiled for Windows on that machine for dealing with all of our data sets. But I would also say that the data have gotten a lot bigger. Back when I was in grad school, you could produce 2,000 tandem mass spectra in a 2-hour run of an LCQ. And we thought that was the most amazing thing ever. Today, you can produce more than 100,000 spectra in the same time on a contemporary instrument. So the data have been getting bigger. The computers have been getting better. And it's been kind of an arms race. But at this point, the efficiencies built into creating software like this have gotten so great that you can do this on a laptop in a real-time demonstration. And that was not possible 10 years ago even. All right, so at the end of the day, we get our report that all jobs are done. And if we click over to our directory, we can see that training run 1 DDA was the directory I created. It's populated with a whole bunch of files. All right, so what is it that we would want to look at here? I would suggest that these I think that these TSV files are probably the most useful thing. Everyone knows what a TSV file is? Tab separated values, not related to me, but the tab characters, thank you for that. Tab characters are used to separate columns here. So you could open this in a text editor, but it's probably better to violate the very first rule of bioinformatics. Does everyone know the first rule of bioinformatics? Don't use Excel. Thank you. Thank you. Good. So, all right. So we're now going to drag combined protein over to our Excel. Yes, I know. You've already converted. I'm very proud of you. Okay. So if I look down this list, I start getting back to something that looks vaguely like biology. I have a list of protein accessions. Here's the short form of them. I have what organism do they come from. The description is here in column H. That's probably useful to expand out a little bit. Because protein profit was used, we have things like protein probabilities to say what is the probability given the evidence that this protein is present. 96% is pretty compelling. We can see some things about the overall amount of data. That's this combined business. All right, so the combined stuff is saying en masse, if we put all 6 of these raw files together, how many spectra do we have, how many peptides do we have, and so on. Oh, thanks. All right, but what we're probably interested in is knowing how our 3-on-3 comparison did. So we have spectral counts here, and I would say spectral counts are a nice quality check on things. They simply ask how many spectra match to peptides for this protein. Okay, so in this case, in the third, in file number 3, which was part of the cohort 1, we had 754 spectra matching to peptides of this particular protein. How are these sorted? They're not necessarily sorted in a reasonable structure here. I might want to sort them by the amount of intensity they explained, or by the number of spectra matched to them. You can use all kinds of different sort orders for this. I think this is like alphabetical or something. Yeah, I'm pretty sure it's just alphabetical by accession. It's not usually a very biological way to look at things. All right, so spectral counts, they're fine, but that's not what we came here for. We're actually interested in intensity information. That's why we chose to do an LFQ MBR operation. Okay, so I have experiment 1.1, experiment 1.3, experiment 1.5. So what this is showing us is that the software has sorted the raw files using the metadata we provided. We said that they were in 2 cohorts, one called 1, one called 10, and then there are 3 replicates in each. It's sorted using that.
It's not so important, but I think we have slightly different numbers.
Yes, there are occasionally some random sampling things that go into these scorings. There are some machine learning routines that are used for deciding which spectra are identified and which are not. So very different random seeds could lead to a change in that. Yeah, if mine says there's a 94% probability and yours says there's a 96, that's well within reason.
Well, it's more about like you didn't change the order of the proteins, right? You just opened it easily. So I assume we have the same order. Like the second row, you have 11, 3, 5, 75. I have 11, 3, 5, 32, 3, 5, 32, 3, 5, 32, 4,
Yeah.
9, 8, etc.
Did you freshly install FragPipe for the class?
Yes.
Okay, so I have a version 22 and you have version 23.
Yeah. Okay.
So there are small changes to the software deployed, and that's well within normalcy, I think. Yeah.
That's why I
Oh, but no, it's worth pointing out. Thank you. All right, so I'm going to color these columns specially because we care about them. Remember that at base, we wanted to know which proteins are different between our 2 cohorts. So proteins are rows here, and these columns represent the intensities seen for these proteins. I just pulled a bit of sleight of hand on you, and I think it's worth talking about. This is an intensity reported for a protein. Did the mass spec measure proteins? No. The mass spec measured peptides. So it has peptide intensities, and that peptide intensity information has been combined to produce a protein intensity measurement. So it's always worth your while to poke into that a little bit and understand how peptide intensities become protein intensities. I believe that this also has max LFQ intensities reported, which should raise some questions for you. The answer here is that some people have gotten used to the way that max quant combines intensity information to produce intensities for proteins. So if you want to emulate that behavior, you would use the max LFQ intensity columns. If you want to do it the ion quant way, you would use the intensity columns. Are the results the same? No, they are not. This is just sort of where we live. This is how things have always been in proteomics. And in this case, it's nice to have the intensity information here. Let's talk about the zero for a moment. What does it mean to say there's a zero value for the intensity of a protein in a given replicate?
Missing value?
Missing value. Okay. Does this mean that no intensity was seen for any peptide for this protein?
I think it means either if the protein doesn't exist or it couldn't detect it.
It could also mean that there's not enough information to conclude that that protein is present, but there could have been peptides from it.
One of the very nice features about ion quant is that it has the ability to detect false quantitations. False quantitations, which means that I might be checking for the intensity attributable to a particular peptide in this raw file, and yet what I see is random stubble. Now I can integrate that noise and give you some very low value, but it's probably more correct to say that this is a false quantitation and thus provide a zero. So is it plausible that some amount of this protein is present but it didn't rise above the noise level? Yes.
Is this noise value sample related? Or is it like a standard, like below certain whatever number it's considered noise, or it really depends on the actual sample you provide?
It depends rather heavily on the sample. If you're doing something like biofluids, the dynamic range of those samples is such a mess that trying to look past the first couple hundred proteins is just really devilishly difficult.
Mm-hmm.
In this case, we're dealing with cell lysates, and that's a pretty favorable environment for looking through a lot of orders of magnitude.
Mm-hmm.
But a given sample may complicate or ease the quantitation of proteins in it.
Yeah.
All right, so it would be possible if we were crazy to make another column in Excel and do a t-test of these three values versus those three values. Please don't do that. It's not the nice way to do it. Instead, we will use FragPipe Analyst to evaluate these data. Okay, so I'm going to close this down. And I'm going to open a web browser. All right. Web browser, Firefox is here. FragPipe Analyst nesvilab.org is what I'm going to use. They have kind of a production FragPipe Analyst server, and then they have the nesvilab one. I often find that the public one is getting hammered while the group one is very quiet, and I think this may just be because we're awake before they are. So it's often very useful to use FragPipe Analyst. All right, FragPipe Analyst is our way to interact with the data after they've been spat out by FragPipe. So I can click on Analysis. I note that I use the LFQ type, and in this case, it is specifically a dynamic I keep calling it dynamic, sorry. Data-dependent acquisition experiment, because that's what we did for this first step. And it says we should upload combined protein TSV or combined peptide. We're going to do protein. And we need to upload the experiment annotation file. What is an experiment annotation file? It is the digest of all the information you provided on the workflow tab. So remember when we entered that this is cohort 1 and this is cohort 10 and this is replicate whatever? That was entering the information that goes into experiment annotation. So I can now upload the combined protein file from my search. It was on D drive, GJAM training, cohort versus cohort. training run 1, combined protein, right there.
My browser is really not loading this website. I tried both Chrome and Brave.
Huh.
I put the shields down. Doesn't work.
Did Microsoft Edge work?
Uh,
Yes, I said that, Chris. Okay. I don't remember testing this on Vivaldi. I haven't used Brave, so
no.
huh. Have you tried the production server as well for FragPipe Analyst?
Sorry.
If we find that one, let me see. FragPipe Analyst.
No, so indeed, I clicked that one.
Okay.
The first option.
Yeah. I'm sorry. I don't know why that's not working.
I'll try to change. I don't know if something with the error on. I'll try to do
It may be something like that. Yeah. All right. So we specified LFQ. That was the default. We specified combined protein here. Now the experiment annotation we need to upload. That was in the same directory. Experiment annotation, there it is.
Which one selected? Which?
Experiment annotation. It's that file right there. So we're going to evaluate the combined protein TSV result by using the experiment annotation. Okay. And I'm going to just leave this with the defaults and use intensity as expressed by einquant. Yes. Oh, good, good. Do you know what was the barrier?
No.
Okay.
Patience.
Okay, so let's go ahead and hit run on that, and hopefully we'll be off to the races. If you immediately get an error, it's probably the case that you uploaded combined peptide instead of combined protein. They look very much alike each other, so it's frequently the case that I click upload one rather than the other. Oh, yes. The FragPipe outputs will be wherever you told it to go on the run page. So on run, you can find this path. That's where it should have output everything. Oops. Okay, so we now have our table. It's probably been sorted by the adjusted p-values, etc. Does everyone know what a volcano plot is? Have you all encountered these? They sometimes give statisticians a bit of a fit, but we'll try to overlook that for just a moment. This is a volcano plot. It's easily the most common way that folks in microarrays or proteomics ask what differs between a pair of cohorts. So we have log base 2 of fold change. Some people are comfortable with log transforms, others are not. So I'll just say that if you have an equal amount of this protein in either cohort, you're going to have a log fold change on 0. So proteins that have no difference between cohorts fall along the center line. Proteins that are way up in the 1 cohort are going to show off to the right. Proteins that are up in the 10 cohort are going to show up in the left direction. How high a protein shows up on this plot is based on its adjusted p-value for being a difference between these two. Now, I told you don't just generate a bunch of t-tests in Excel. One of the reasons for these are the abundance of those zeros in the table. Having a bunch of zeros there will not do you any favors in the t-test because the zeros look like they perfectly replicate, and so they have no variance within a cohort. That's a real problem for artificially boosting the negative log of the p-value. So one of the things that this is doing is imputation. Does everyone know imputation? Okay. When you have a missing value, let's say when you tried to integrate a protein for this raw file and you couldn't find any of the peptides, you have a 0 in your table. So FragPipe Analyst knows that 0 is a missing value and not a measured 0. Measured 0 versus missing value, everyone get that? Okay. So the software is using imputation to fill in some value randomly selected for that missing value. The model that it uses for this can be configured under advanced options. I tend to leave it at default, but your mileage may vary. All right, so now we have dots that represent the proteins that have been identified if they are high on this list. Well, if they're, I think it's not actually showing a line across here, but if they have a sufficiently low p-value, they are sufficiently high on the volcano plot. So things that show up here are significantly up in the 10 cohort. Proteins that are up and to the right are proteins that are up in the 1 cohort. That's a little weird, isn't it? Because we know that both of these are yeast background with more of the human proteins spiked in to the 10 cohort than in the 1 cohort. So we wouldn't really expect to see any proteins that are up in the 1 cohort and down in the 10 cohort. That's a little confusing. So we can investigate why each of these shows up. And it would be worth our while to be able to color differently those ions that are those proteins that came from the human database, because the human proteins should all be on this side, because this is the side favoring the 10 cohort. All right, so that's a brief look at how to discuss a volcano plot, but it might be that you're particularly interested in just one particular protein. So let's highlight this top one. I'm going to click the top row. It'll re-render the volcano plot, and now it's going to put a little flag on where this protein shows up. So you see it's shown here in magenta. That's our protein that's differential. Its log base 2 fold change is negative 6.51. Now, I would need a calculator to figure this out, but I would need to raise 2 to the negative 6th power in order to figure out the linear fold change of this protein between cohorts. That's more than we would expect. 2 raised to the 6th power is like 64. Is that protein 64 times as abundant in the 10 samples as it is in the 1? No. That protein should be up tenfold. So there's some bounce in the ratios estimated by these things. Occasionally you're going to be too high a ratio inferred for a protein than is real in this case. And it works the other way. You might have a protein that you know is 10 times more abundant that gets measured as 4 times more abundant by the software. But I can also do useful guides by looking at, say, a feature plot for a particular protein. Now, this is the most differential protein, remember? So we have these 3 measurements. You can see the dots right there. The box is not all that informative when you've got just 3 points. But we see that there were 3 measurements within the 10 cohort. But I only see 1 dot over here, and that's a little concerning in the other cohort. Let me show imputed values. Okay, now when the software came up with a p-value for this comparison, it had to do something about the zeros that were reported. And so we see that these are the 3 values that were used for it, and they're all positive at least, so it's now got some sort of imputed value over here. That's where it came up with its negative 6.5 log 2 fold change. But it's useful to be able to show real data versus imputed data for a protein, because when you have a really extreme p-value, it's very likely the case that at least some of the points on one of the cohorts is imputed.
Yeah, for the imputation, what would be the best way to do it?
Oh, I'm simply going to say there are people who understand imputation very well and people who understand imputation superficially. And I'm probably in the superficial category. I know that if you have to rely on imputation, you're already in kind of an awkward place. I don't like having to rely on it, but we're always going to have missing values in quantitative proteomics. You can see that there are 6 different methods used here. Why would it be bad to impute zeros? To say if there's a missing value, I'm just going to take it as a measured 0. Again, there's no variance in it. So if you have 3 imputed zeros, they look like there's no variance within one of the cohorts. It does bad things to t-test. If I use k nearest neighbors or MLE is maximum likelihood estimation.
I think so.
Yeah. Or minimum. This is actually kind of a popular route for some things. If you simply said, what is the lowest intensity I've seen for this protein anywhere, I'm going to impute that everywhere where I have a missing value. Again, you don't have any variance. So that's also problematic. There are lots of ways to screw up imputation. I tend to stick with the default because this is the way the field does it, but there are some limitations to that.
What about the normalization of the data? I see that it's not normalized.
Yeah, that's a tricky one. Samples from complex studies are frequently collected over multiple days, and you may even have multiple batches in play. And if that's the case, you really need a model more sophisticated than what FragPipe Analyst is going to give you. If you had quite variable response from the instrument, maybe let's take an example like I was cutting bands out of a gel, and maybe in the one case I got a bit more protein harvested from the gel band than in the other, then I'm probably going to have some sort of normalization to adjust for the different quantity of protein. This is not something where I have a hill I would die on. It's an area of the data analysis that having a proper biostatistician rather than somebody who kind of talks about biostatistics would be helpful. And frequently, a lot of the projects we were doing at my prior university, we spent a lot of time trying to find anybody we could talk to address the biostatistical issues. The bioinformatics were not necessarily the limitation, but the interpretation of the values from bioinformatics were very much the issue. This is one of the reasons why I ended up teaching myself how to do statistical models for multi-factor tests, because we just couldn't find statisticians who were equipped to help us in proteomics.
But when you do have to perform data normalization, do you have a certain method that you prefer, or it varies per experiment?
In this case, we have three options. I prefer massaging the data the least necessary, so the default of no normalization is already where I would leave things.
Okay.
But if I knew that the instrument had very variable responses across the experiments, I wouldn't have an alternative to using normalization. Yeah.
And does FragPipe or the analyzer provide any sort of visualization on the data for you to make a decision whether you want to normalize or not?
Honestly, the number of visualizations within this are pretty extreme. Something like the heat map could be quite useful for that. If you know that one cohort is systematically more intense than the other, then something of the sort's going to be needed. We ran into that a lot with affinity proteomics doing pull downs, because negative controls have nothing like as much protein in them as do the positive experiments.
PCA would also show if there is an outlier, right?
It can be, yes. I believe that's included at the bottom here. So if I roll down a bit, I get to PCAs of the samples. And in this case, I mean, they look, they're spreading all over the place. So, yeah.
Sometimes you see one sample will be really out of the group, so.
Density plots would also be useful.
Yeah. Finding outliers is an interesting problem to work on, and something I would often do from just quality control data rather than identifications, but you can do them based on intensity tables too. Yeah. Okay. So.
The density plot might also be useful for this. The last one in the QC plots.
Oh, here it is, yeah.
Because it will sort of give you an idea of how well they overlap and then how big of a chunk is imputed.
That's true.
So that could also be.
Yeah. Yeah, I won't be walking through what all the visualizations are used for, because I don't use them all, but I'll just say that at a glance, if you're trying to make a model where you're comparing one cohort to another, FragPipe Analyst has a lot to offer you that is already in place and doesn't require you to script up a bunch of R yourself. The imputation step is the one that I find most daunting. So that's my personal demon. once you've done an imputed table here, you can export it and then work on it in R as well, which is quite necessary when you're doing multi-factor models. Okay, I'm going to close this down and come to the other example. Leave the page. All right. So we've now looked at an example of a DDA experiment. So data-dependent acquisition means that we collect tandem mass spectra for intense peptide ions. The case of DIA is quite different. DIA collects tandem mass spectra on a schedule, and it doesn't expect kind of the one peptide, one MS-MS expectation. The idea in a DDA experiment is that each MS-MS is evidence for one particular peptide. But once we get to DIA, we're collecting with a broad window of precursor ions, a range of different mass to charge values that we're accepting all peptide ions within, co-fragmenting them, and then reporting their fragments out as an MS-MS. The chief advantage of this is that it gives us multiple MS-MS measurements for peptides. We should be able to recapture the MS-MS for a given peptide 2 or 3, but ideally more like 10 times over the space of a chromatogram, which means that we don't just have the rise and fall of the intact peptide ion in MS1 scans to quantify on. That's what all that business was based on. Instead, we can look at the rise and fall of fragments attributed to this peptide as a function of chromatographic time. That means that we can quantify it by multiple channels at once, rather than just relying on the peptide ion rising and falling. Now, the data that come from this are a lot more complex as a result. But helpfully, FragPipe is able to identify them pretty much as straightforwardly as we did this set. So I'm going to return to the workflow page. I want to clear away all the files that I had before, and I want to change the type of workflow that I'm going to use. I'm going to use the DIA speclib quant workflow. What this means is that our identification algorithm in this case is going to attempt to build not just a set of peptides that can be identified from the spectra, but also associate them with which fragment ions do we see for those peptides as well. So this combination of a list of peptide sequences and the spectra that associate with them is called a spectrum library. If you see this, it means this peptide is here, in short. The quantification step is then to use that spectral library and ask, what intensity would we attribute to each of the peptides in this library that we just built from the data? All right, so once again, I want to add the files. But instead of the DDA MZMLs, I have to expand out the other zip now. Okay, here it is, the DIA MZMLs. That's the file that you copied from the thumb drive, and now we want to extract those. These are larger. They are larger. DIA experiments tend to collect far more spectra because they are collecting redundant spectra. Oh, did the laptop melt down?
Hopefully.
Ah.
If these spectra are larger, they require more RAM to run the analysis. He already ran out on the DDA. You have no chance now.
No, exactly.
So sorry. Okay. Yeah, there's always the risk that the laptops will not measure up to the moment. The worst part is when my laptop does that. So, yeah. Okay, so now we have new files. These are each, well, let me see. Originally, the DDA ones were about 3/4 of a gig apiece. These are a little bit more than a gig apiece, which is why I gave them to you as zip files. All right, I return to FragPipe. I want to refresh this directory. I want the DIA MZMLs. I select those. Once again, I need to decorate them with cohorts. This is experiment cohort 1. This one is 10. This one is 1. This one is 10. This one is 1. This one is 10. And again, I don't care too much about what their replicate numbers are. Does anyone spot something that I've done wrong already? Yes, over at the right, it calls these DDA files. The software allows you to combine the analysis of DDA and DIA files. So I need to highlight all of those and specify these are all of DIA type. Okay. Did your software automatically flip them over to be DIA by default?
Yeah, also DDA.
Ah. So yes, select them all and then click Set DIA, or you can do them one file at a time. Either way. Okay, the database is unchanged. The DIA sudo ms2 step is still disabled. That should be. All right, MS-Fragger, it's really going to be run very much the same way. Validation still needed, of course. PTMs still not in play, no glycosylation. We are no longer running ion quant. You see that this is now unchecked. This was the effect of selecting the speclib, the DIA speclib quant workflow. This turns off because this is no longer the way we'll quantify. Nor are we using the isobaric quant, but we will use a spectral library. So we can see that it's going to generate that from the search results. And then we look at quant DIA, we see that this is turned on, again, because changing to this workflow. So Diane is going to be used to produce intensities for proteins from the DIA data. All right. Skyline is still turned off, I believe. But now I need to specify a new place to store this result. So I'm going to pop out of there and call this one training run 2 DIA. Okay. I'm getting very, very original. Now, I don't want to be confused by any of the old stuff that was on screen, so I'm going to click Clear Console and delete the temporary files. And then I should be all set to run. I'm going to go ahead and click Go because this takes a bit to run. All right. And pretty soon we should see memory and CPU usage skyrocketing upward. Yes?
For the DN configuration, do you know which default parameters FragPipe will use? Because with IronQuant, we activated the MBR.
Yeah.
DN can also do that.
Yes.
But you have to specify what it should. Do you know if FragPipe does that?
The easy way to check would be to do the dry run option over at the left, because it will show you exactly which commands will be issued along the way.
Yeah.
But it seems like a very obvious thing that must be turned on. I'd be surprised if it fails to do that.
Yeah, it's just that like if it's turned on by default, then there's no way to turn it off. There is not a button that says MBR in the DN quantification window. But there is a command line options line.
Yes.
Where you could put, in DN I think it's called reanalyze.
Yes.
So you could put that in, and then it would do MBR. But if you don't put it in, it won't. And so if it's already on by default, there's no way to turn it off.
Yeah.
That makes sense.
When I was doing a paper involving Diane last year, we had data where we hadn't used iodoacetamide. We had instead used MMTS. So instead of gaining 57 mass units on the cysteines, we gained 46. That was a real struggle because getting Diane to recognize plus 46 mass shifts requires a command line option, not just specifying it under MS-Fragger.
Yeah, and
that information wasn't
good.
good.
Like R mod, unique mod, whatever number with mass change and location. Yeah.
Yeah.
Convoluted. Yeah. Sure.
So I didn't do the dry run on the run tab, but that should show exactly how it's invoked, and if it's failing to invoke something it should, that would be worth knowing.
Yeah. So it was just I would hazard a guess that it does not do MBR.
Really?
Yes.
That would be very surprising to me.
But it should tell you in the output then at some point it will get to like it will do a reanalysis run in the end if MBR is active. And then you will see that. If it doesn't do that, it's because you don't have the reanalyze flag attached. And it would actually almost surprise me if it was on by default because you cannot turn it off then.
I see.
And there could be situations where you want to turn it off.
Well, we will try to spot that in the output as it goes.
Yeah, just a heads up, this might not do MBR then.
It's possible, I guess. As I said, it seems like something that so clearly needs to be included.
Yeah.
Do you mean that it wouldn't attempt to do time warping to make the
No, it would still do like RT alignment.
Yeah.
Yeah, and it would still do all of like the predictive models, but it would not go through all of your output again with the new spectral library that it builds as part of the
Yeah.
source run.
Yeah. On the quant DIA page, I wanted to see where IRT gets called into play. It doesn't get included here.
It's the quant DIA page tab is limited.
I would agree.
Put it that way. And so unless you know all the command line options, you don't really stand a chance. Hence my question.
Yeah, the devil is often in the details. Shifting to just a different way to handle cysteines creates a bit of a nightmare for adapting the software to that purpose.
Yeah.
I ended up having, like anybody else, going to the online forum for FragPipe and posting an issue. And they got back within 24 hours on how to do
Oh, yeah,
this.
really good.
Yeah. The support within the FragPipe group for actual users is stunning. It's got to be a good fraction of the postdoc's time to keep up with that stuff.
Oh, sure. What's his name? Feng Chao.
Feng Chao.
Yeah,
Feng Chao.
He spends a lot of time on GitHub.
Yes, he does. Yeah. Okay. So we're having a rather different experience now. We still did our pre-search at the first just to figure out the mass calibration stuff. But look how much longer it's taking on each MZML file separately. We see that the experiment has 120,000 MSMS scans, and even though it's clocking along at better than 2,000 spectra a second, it's taking a fair bit of time to analyze the identifications. So let's spend a moment then on the difference in MSMS spectra for DDA experiments and DIA. In DDA, each MSMS is triggered to sort of take a snapshot of this particular peptide ion. It had a big signal in MS1, therefore we think we can get a high quality MSMS by targeting something at that mass to charge. So this MSMS should represent a bunch of ions of this particular precursor mass to charge breaking. And we're measuring their fragments in the MSMS. In DIA MSMS, each MSMS represents a set of peptide ions that are co-fragmenting. So our individual MSMS, we're not trying to say we need to account for everything we see in this MSMS with a particular peptide. Instead, we expect that multiple peptides are sharing this MSMS. And we expect that through time, we're going to have multiple MSMS scans attributed to this. So when you see, I guess it's going to be after this stage, something called the DIA tracer going on, it's partly trying to stitch together this time dimension for the fragments and the precursor ion that represent a given peptide. So let's imagine that we have two peptides, and these peptides overlap in chromatographic time, but they do not, they're not identical. It's not like when one peptide shows up, the other does, and then they go away at the simultaneous time. Let's imagine that one shows up 15 seconds after the other. So I have the 30 seconds in which I can identify and maybe quantify peptide A, and they overlap by 15 seconds with the 30 seconds in which I can see the ions of peptide B. So if I have multiple snapshots during that overlapping time, what I see is peptide A going away, but peptide B coming on strong. It's coming up from nothing to be a very dominant ion. So the process of deconvolving that, of being able to say that the signal for peptide A has already hit its maximum by this time and it's fading away, and the fragments for peptide B are coming on strong and then they'll fade away later, is all part and parcel of this quantitation problem for doing DIA. And within the identification step, we need to be able to recognize that multiple time slices, multiple MS scans for this set of precursors are giving us multiple snapshots of the same peptide's evidence. So where in a DDA experiment, we expect each MS to be our only line of evidence for some particular peptide, in DIA, we really need to take account of the fact that the fragments for this peptide are observed multiple times. We can use that for things like noise cancellation to say this fragment ion is observed in this MS, but it's not seen in the scan before it, and it's not seen in the scan after that. Therefore, it doesn't matter as much in identification. It's more likely to be noise, or some peptide that just is a flash in the pan that shows up and then vanishes. This means that we have a much larger collection of tandem mass spectra to work with because of the rate of acquisition. But it does mean that the evidence for a given peptide appears across multiple MS scans, and stitching that together is work. Trying to think what other things I would mention about these along the way. I would say that DIA data analysis is actually really pretty new for me. I was in the camp of people who said that you could never identify as deep a proteome in a DIA experiment as you can in a DDA. And the field has proven me completely wrong on that score. DIA, by doing multiplexing, each MS representing multiple peptides, because it's multiplexing the evidence for different peptides into MS scans and acquiring more MS scans, covering far more of the available space, you can get a deeper proteome from a DIA experiment in the same instrument time that a DDA experiment takes. So that was a big surprise for me. But you do pay this awful cost. The identification time goes up.
My computer needs
Huh?
You don't get like a fingerprint spectrum.
Right, yes.
That you get from DDA.
Yeah, the software is going to attempt to reconstruct that. The stuff that goes into the spectral library will be what fragment ions were measured for this peptide in the DIA scans, not just the raw DIA scans as they are. So there are lots of tools you can use for this. We're doing something called library-free data analysis for DIA. We didn't have a pre-existing set of peptides and spectra that we expected to see. So that means then that we have to go through this process of identification, and it's costly. If we were to do the same experiment in Spectronaut, Spectronaut would be using its search engine Pulsar to achieve the same thing. And Pulsar was designed for DIA spectra in the first place. MS Fragger was originally written for DDA spectra, and it's been adapted to work in DIA as well. And of course, the software Diane is also able to do searching of spectra to do this identification step. The way that FragPipe deals with DIA data and the way that Spectronaut does it, or that Diane does it, differ in some important ways. We don't know all of the ways in which they disagree, because Spectronaut and Diane are not ultimately open source. So we don't have the ability, and frankly, you could make the same complaint about FragPipe. It's available under license, and yes, the source exists. But it's not all that open as source code goes. So do we know that the way that these three approaches compute false discovery rates are really comparable. Do we expect that if I identify a peptide with one of these engines, it should be identifiable by all of them? There are some important differences here. What I'll say is that FragPipe, in dealing with DIA data, tends to be relatively conservative. The inventory of peptides that it produces is, on the whole, I would say, more confident, more trustworthy than the other algorithms. On the other hand, if you see a huge increase in the number of peptides you see for a sample in Spectronaut, the odds are pretty good that you can rediscover those analyzing the same data with Diane. So if Diane and Spectronaut say the peptide is there and FragPipe doesn't, I guess I would put it down to the grumbly bioinformaticist like me, who says, well, that result is too good, I don't actually believe it. And yet, the information from Spectronaut and the information from Diane has proven to be pretty trustworthy. So I would not be one to cast aspersions on that. Having spent a year just trying to evaluate whether the additional information you get from your data are trustworthy in the case of affinity proteomes. Chris, what do you use for DIA analysis most typically?
It depends on the purpose. If we're looking for something that will produce like pretty figures out of the box, we use Spectronaut. Because Spectronaut has a robust visualizer.
It really does.
And
very nice QC tools.
Yeah, very much so. If we're passing the data on to you and G and they will do the downstream, we typically do DM. And then you essentially get tables of information, and then you do the statistical analysis. You can do that in FragPipe Analyst, but there are also lots of other options.
Yeah.
And Ewan does most of his work in R, I think, directly, right? Yeah. So it very much depends on the purpose.
Glad we have you here. Our folks who feel really comfortable and happy in R are valuable people to know. Definitely. Still working on it. I have to say, there's this challenge when you're doing a training and you are recording video on the laptop as you're running the software on it. You end up using a processor core just for recording the video, and cuts into the performance time of it.
Consider whether you want to do the TV kitchen thing of a pre-baked one.
Yes. Yes, we frequently do that. When we taught a demonstration on top-down proteomics recently, we provided them the outputs that they would get from, if you ran the search on all 6, this is what it would look like. It's going to make it, though. So if we were to try to reconstruct how we got to our result in FragPipe the first time, we would say we did identification. We matched peptide sequences to tandem spectra. We did filtering and assembly. We decided which PSMs we trusted, which peptide spectrum matches we trusted, and threw out the rest. And then we inferred a set of proteins to go with it. And then we quantified, basically integrating the speed bump we saw in the MS1 scans. DIA is not so very different. We still have our identification step for where we allow for each MS to tell us about multiple peptides and multiple MS scans seen for a given peptide, which is why it's taking so long. We would also do filtering to decide which of the identifications are actually credible. We export a spectrum library, which is a list of which peptides we believe we really did see, along with which fragments they should produce. And then we're going to quantify on that basis. The first 2 steps are the slow ones. And then quantification is going to rocket by very quickly. I swear file 6 was taking much longer than I thought it should. All right. Finally moving a bunch of files. The step of identification is complete. Yes, sir.
I would make a more mass spectrometer question.
Okay.
Do you have suggestion on the optimal data points per peak.
The optimal.
Optimal data points per peak for the quantification.
Number of peaks to like, characterize peak.
Yeah. Most of my opinions on this came from Mike McCloss. We crossed over when we were back in Yates Lab many years ago. If I had to pick a minimum number of peaks, I would probably say something like 7 measurements across a chromatogram is quite necessary to getting a decent peak shape, but I'd much rather have a dozen. When we created our variable windows method for DIA collection on the Explorus, we said we can generate an MS-MS every 10 times every second. We can see a peptide for about 30 seconds. So if we want to get 10 measurements within 30 seconds, we need to complete our cycle of DIA in 3 seconds. So we do 30 different MS-MS scans then in each DIA cycle, 30 different windows, which means that we're getting 10 measurements within 30 seconds for a given peptide.
That is a lot, yeah.
That was our goal. I don't actually know how trustworthy the rule that a peptide shows up for 30 seconds is, because you frequently see very different measurements estimated by.
The median value, let's say, some will be with very few, some will be with more, I guess.
Yeah.
Okay. Yeah.
So that was our rationale with the Explorus. Would that same method be equally applicable in a timsTOF? No, because the scan rate's so much higher. You can get away with a lot more isolation windows than you can on an Explorus. And of course, you have the option of building in ion mobility separation as well.
Yeah, this is what I'm working now in timsTOF. And now I have it in like 6 data points per peak, but
I would like better than that, yeah.
okay.
The spectrum will be happy if you have 3. So.
Yeah, this is why I'm asking, because I see that now the minimum required number is getting lower and lower.
Okay, so does everyone understand what this question's about? All right, I have a peptide showing up and then fading away in kind of a log normal shape, I guess. If I have 3 measurements of intensity within those 30 seconds, what is my ability to quantify it? What is my ability to make an integration of that shape that bears any relationship to reality?
Poor. Yeah.
Very poor, very, very poor. If I have a sample showing 0 intensity just before, and then a quickly rising intensity, and then I can track its fall off with another 10 measurements, that's brilliant. But the more times I sample this, the fewer the number of isolation windows I can consider. So there's always going to be a trade-off in our method developments to try to keep the windows of isolation narrow enough that we're not trying to quantify 100 peptides at a time in a single scan, but see them frequently enough that we can get time resolution on its intensity profile. Okay. Hey, we seem to have made it over into Diane. This is good news. So we end up using Diane twice in the FragPipe workflow. Diane is first going to be used to help us to gauge our confidence in a given peptide. That means that Diane is going to be used to predict what fragments should we see for a given peptide, and at what retention time should we see it. So it's doing this process right now, but it's got a pretty large number of ions to do that for. And there's no way to say it other than that Diane is a CPU hog. It tends to just park you at the full wattage your CPU will bear and just keep there for a while. So we're going to take the moment of predicting what the spectra should look like, and then it will predict what retention times those peptides should show up at, using that to help us build a spectral library that's confident. What is the cost in the end? If the software claims some protein is present, but all these peptides are actually false positives, why is it problematic to have that in a spectral library used for quantification? The short version is that the spectral library represents the set of peptides that we are going to then go quantify in this DIA data set. If I gave it a bunch of random peptides that don't really exist, but I put them in my spectral library anyway, the software will dutifully come back with some quantification value, some intensity value for ions that it sees for this non-existent peptide. So having a larger number of peptides than are really present is a bit problematic for that. It's tempted to go look for quantities that don't exist. Ultimately, when you're making your comparison of this protein is or is not differential between these two cohorts, you would like that list of proteins to be relatively compact. You don't want a whole bunch of stuffing in there of peptides and proteins that don't really exist. So the software is being careful about building this inventory, and the inventory will then get shoved into a spectral library. Yes, retention time regression, blah, 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 blah. So in this case, we have the experimental problem that liquid chromatography, as used in proteomics, is a pretty approximate separation. Is it possible that the column goes gloop at some point and you end up with a bubble? Yes, that can happen, and it makes it harder to compare the intensities we see from this experiment to the ones we see to this one. Is it possible that the pump is a little irregular, and so the gradient is a little higher in overall hydrophobicity in one experiment than in another? Yeah, that can happen. So we want the ability to say that a peptide seen at this retention time in this experiment is the same peptide as was seen at this similar but different retention time in another experiment. That's what retention time regression is all about. I did mention this stage would take a little longer than the DDA experiment. So why would we go about changing it all up? I mean, for 2 decades, everyone in LCMS/MS was doing DDA. Why would DIA suddenly eat DDA's lunch? At base, the quantity information that we used in our first experiment in the DDA analysis was built almost entirely on MS1 data, okay? When we had a particular peptide we'd identified, the quantity value that we used for it was all drawn from MS1 scans to say this peptide ion had this intensity, then it had this intensity, then it had this one, then this one, then this one, then this one. But that's all one ion that we're evaluating, or at best, we see the same peptide at multiple charges that have several isotopes of it. Maybe we combined information from all of those, but that's a fairly limited base of information for building your quantity upon. DIA is very powerful because it allows us to quantify not just on the basis of intact peptides, but on the basis of the fragments that result from them. We have a chromatogram for the precursors, just as before, but we also have chromatograms for fragments. That's a broader, more stable base of information for us to base our quantity of this peptide on. Okay, have we finally made it to protein profit? Or is this percolator? This is percolator. We're getting there. We will soon be writing a spectral library. I promise. At least it's only 6 files, right? It is, however, 5 minutes to lunch. I have strong feelings about lunch. And I will not judge anyone who walks out of this room with a spot of noon.
I know, I want to see it.
We've wasted so much time getting to this point. Did you ever take an economics class? That's called the sunk cost fallacy. the sunk cost fallacy. You've invested so much effort in getting to this point that you would willingly cut into your lunch hour in order to see the result. The result is better pay off, is all I'm saying.
It doesn't justify economics though.
Yeah. All right, so I'm going to mention one more thing that's probably useful to everyone on this. How would you judge that the quantification system was working? I would say one of the simplest things to do is to shoot the same sample a bunch of times. Maybe you shoot them even on different days, because instruments have different opinions on different days. Seriously, they really do. So if you shoot just a bunch of yeast guts on the instrument, time and time again, shoot 10 replicates of the thing, you can then ask, what is the coefficient of variation that I see for measurements of this protein over time? And I can tell you, one of the things you're going to see is that proteins that have high intensity, that produce really loud peptides, tend to have relatively stable quantitation, but proteins based on peptides that have very minimal signals tend to have a lot more bounce in them. So evaluating this relationship between the coefficient of variation and the overall intensity for a protein can be very useful, very instructive about how much you should trust any one measurement. And by just shooting the same sample lots of times, you will know that the actual variation is 0. So that's very helpful for making that kind of assessment. All right, it is finally grabbing all this stuff together to build us a spectral library. And we see that the CPU is jumping again. Do you see this business about rank 1, rank 2, rank 3? Like right here? This is to say that the software has attempted to identify the MSMS scans in a way that says, my best guess of a peptide that relates to this MSMS is this peptide. That's a rank 1. It then removes those ions and says, what's my second best guess for explaining what's left? That's my rank 2 peptide. So you can see that it's keeping up to 5 for each MSMS. Any peptide that doesn't make it into the top 5 of at least one MSMS is jettisoned. So that may represent a difference in how Pulsar or Diane would go about identifying than this does.
When you're then talking about an MSMS here, are you talking about the actual recorded MSMS or like an extracted DDA style a single precursor MSMS?
Oh, yes, this doesn't do that. We did not use the IA amplifier.
No, I know that's because if you only get up to say 5 ranks from a single MSMS scan, that would mean that your entire window can only comprise up to 5 precursors.
Not really.
No?
No, because you have multiple time shots. You have multiple samplings in time.
Oh yeah, no, but like for an individual run at time whatever seconds, there's one MSMS scan in this particular window, and you can only find 5 precursors.
Yes. For a peptide to be detected, it must be in the top 5 for at least one MSMS.
Okay, but they are recorded still in the entire period. They're just not necessarily like in the top, but you could still use the other measurements for quantification. Do you see the issue there? Like, so if I have a protein, peptide, that I detect as part of my top 5 once.
Yes.
But I still need the 12 data points in my chromatogram to quantify based on it, right?
The quantification step is not where we are, though.
No.
We're just building the spectral library.
Yeah, okay. So
If it makes it into the spectral library at
then
all,
you will quantify later on.
we'll attempt to quantify it. Yeah. This is just to say that in order to get identified in the first place, you've got to be at least in the top 5 of one. Okay.
Then not a problem.
It's 11:59. Really, no judgment. If you are unable to see it, we can look at the result.
We'll launch it till next meeting.
Yes, I understand.
Thanks.
Thanks for coming. Okay. We've now parsed the information, converting it, blah, 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 blah. We're going to get there, I swear.
Yeah, for me it's, I don't know, 10 minutes or 15 minutes ago finished. In 20 minutes.
Oh, yours finished already? We should plug her into the screen. Your total run went 20 minutes. That's very nice. Yep.
Congratulations.
Yes. Yes. I was compressing H.264 video at the same time as I was doing this, so yeah, let's chew into it. I can speed it up. To save further time, I would suggest that we come back to see the result of this after lunch. So we're back in here at 2 o'clock for PTMs. If you're not able to be here for that part, I understand. But I will include the link for you guys pretty soon.
Okay.
I thought you started to say something.
No, I said okay, that's nice.
Okay, cool. So yeah, I'll probably just put this on YouTube. And
Yeah, that would be. Thanks.
Cool. So I'm gonna go grab a pizza.
From where? Domino's?
And probably just Domino's, yeah. But yeah, I will. I may go ahead and just keep recording video until this is.
Oh, thanks.
Cool. Yeah.
Yes.
Thanks for coming.
Oh, of course.
Thank you. I am recording it right now. Yes. Thank you. I have to use these a lot, so I will worry about getting them back. Thank you. Decision to record it. Oh, well.
You can always change your mind about posting it later, right? Can be one of those people who decide to post it, but stupid they do and then post it online anyway.
Yeah, the last time I recorded the class, I recorded the whole thing in video and then realized I had my microphone muted. It killed me.
No.
Yes, it happens. Thanks, guys.
Merging file or which step?
It still has not exported the spectral library.
Okay.
It's just taking its own sweet time to get there. I need to update my laptop again. Thanks.
Have a nice day.
Thanks. Well, for those listening to the recording, I'm still here. And we will sit through until this starts the quantification step, at least. I would point out that the FragPipe Analyst software looks very slightly differently when we are using data from a DIA experiment than when we're using a DDA. So the LFQ option is what we used the first time. And you can see we gave it the combined proteins file and the experiment annotation. But when we use a DIA experiment, we need to specify that. You can see there are two options for DIA. There's the DIA for proteins and DIA for peptides. To keep things as comparable as before, I'm going to say use the DIA option. And instead of uploading combined protein TSV, we're directed to install to upload the report protein group matrix, PG matrix. So we're getting an altogether differently named output that comes from the FragPipe software. And any day now, it will generate a spectral library. Oh, you see, we're actually in the spectral library generation step now. So that we've completed identifications, we've completed filtering. Now we need to write a spectral library that we trust. Now we see that the software is going to regenerate is going to redo this Diane step using just the protein groups and precursors that were found at the first stage. Thank you for saying, Chris. I appreciate it.
No worries. I was looking at your DN sort of startup message there.
Whether it's using the
Because it is not, but I don't think it's a problem. Now that I think about it a little bit more. So you can see there's no line that says that match between runs will.
Yeah, the match between runs was invoked. It wasn't.
No.
Interesting.
But I don't think it's a problem because you are already generating a spectral library from your input data with MS-Fragger.
Yeah.
And so it uses that library during its identification run. So I don't actually think it's a problem.
Yeah.
It's just something that's interesting to keep in mind.
Yeah.
Because I tried to put the reanalyze flag on in the command line options, and it actually broke the pipeline.
Okay. So I think that's pretty clear. It's not doing that.
Yeah.
By the way, this is the quantification step actually running. So the library has been generated now, and it's being applied in this quantification step. And I think I will not be the only person who is surprised by how rapidly the quantification runs compared to all of those other steps.
No, the quantification itself is quite fast.
Yeah.
I think I have a log file somewhere.
Yeah, it's worth noting that FragPipe isn't just going to show us this information as it runs on the GUI. It's also writing it to a report log file. So that should show up along with everything else in the report. We can look at the run that we did earlier. And if we pop down here, we see a log file, the date it was run, and so on. This is really helpful when you're writing up the instrument methods for your manuscript, because you'll probably want to know which version of the software did you use and how was it parameterized and so on. All of that information should show up in here. If you just want to know how was the search run, you can look at the Fragger params file. It's just a big text file that shows how all those different options were set. As we come back to this, though, we see that we're closing in on the very end of the quantitation. Just a minute apiece. That is really nice. Yep.
Fortunately, that's the DN library search is pretty fast. But if you did this whole thing in DN directly, not fast.
Not fast at all on DN, yeah. No. On this laptop with my sturdy 8 cores, yes, it would be problematic.
Yeah. As you can see in my log file, I have MBR enabled here. Which is because I put in the reanalyze flag.
Yeah.
But if I then go down to the bottom, it finished the main search and then the reanalysis search, but then it's supposed to move some files around and it can't because the names are different.
That's vexing.
So because I get these first pass output files followed by the main PG matrix, etc. So it bugs the full pipeline. So I don't think you're supposed to put it on.
Okay. Well, we've done 2/3 of the quantitations. We're closing in on the end. Soon we will have our report PG matrix.
13.9 minutes, but I'm cheating.
13.9 minutes.
I dumped it on the new processing unit.
Yes.
Over in the basement. That seemed a little bit.
Running this on a desktop is a very different experience.
Running it on a server with 128 cores is a little bit different.
Yes, it is. Yeah, I'm typically running on a 12 core at home, and its ability to rocket up to a high clock rate is wonderful.
Yeah.
I don't think any of my computers is below 32 gigs of RAM at this point. It's just like I run the software often enough that I want to benefit from it.
I think this one might actually be.
Okay, and we're quantifying the last of the 6 files.
Took 10 minutes extra.
I'll have to be careful to make sure that this stays on time for the GJAM session, though.
Yeah, I mean, I would do the TV kitchen thing of having a pre-baked one just in case.
Yeah.
And then
I can add that to the image, no problem.
yeah. You can still, run this in the background, but then while it runs, which it will do for half an hour,
you can show the FragPipe Analyst from the prepaid.
Yeah.
I think that's the.
I think I may update to FragPipe 23 for the course as well, since that's what the students would just have installed.
Yeah.
And little differences between how this GUI looks and how theirs looks is, there's no need to confuse the students with that.
No.
Yeah.
FragPipe is now up to 23.1.
Yeah.
Is the last.
I downloaded it this morning, yeah.
Yeah.
So the cross-run analysis allows us to stitch together the data across all 6 files and control for error of that sort as well. We see that there's a report generation that gets kicked off here as well. But often that runs just slightly past the end of the FragPipe run as a whole. There we go. And often it's because of this little error right here. These raw file names are very long, and so creating the QC report for how Diane went causes a tiny little glitch, which is why FragPipe finishes up here, and the report generation is kind of allowed to extend beyond the end. So did it write a report? Obviously that's something we would care about quite a lot. We can come back in here, go to training run 2 DIA, and here is everything, except we still have some of these junk files left around. So it didn't delete all of the temporary files as we would have hoped. But I do have the file I need here. So I should be finding protein group matrix. Let me sort by name. And I want to look for protein group matrix. Where'd it go?
It's in the DN output folder.
Ah, right. Here's Diane output up here. Here we go. Report PG matrix is here. So that's still in place. Do I still have my experimental annotation file? Oops, sorry. It's right here. So we're in a good place. We can simply browse to the Diane folder to grab our report PG matrix. Not GG matrix, the PG matrix. That's protein groups. We can grab our experimental annotation as before. That uploads, click run, and very shortly we should be presented with the same kind of plots that we saw before. The number of proteins is likely a little different because you can get a lot more identifications out of DIA if everything is going well. And we see that 32 proteins are called differential here. And one could go through to check each of these protein IDs that are called differential to ensure that they are coming from the human database. But I will note that in the DDA experiment, we had a whole bunch of proteins that were called up in 1. Those have vanished. I think that the improved quantitative stability of DIA probably helped it to realize that these proteins don't exist. There are no proteins that are higher in the 1 cohort than in the 10 cohort. And here all these magical proteins are. A great many of these should be proteins from the human database, representing the proteins that were spiked in at 10 times the concentration for that cohort. And that concludes the first demonstration of the day. Thank you for lingering past lunch. And we will now shut this down for and wait until the start of the second demo. Well, it didn't actually stop.